Welcome to my channel for NEET Biology. Today we shall discuss about the linkage and crossing over. In the previous classes we have already discussed that what are actually the incomplete dominances, what are the inheritance of traits, and what are the codominant traits. Today we shall discuss about the linked genes. What are the linkage phenomenon? What are the crossing over? What are the criteria of linkage? That is, why the genes or factors of Mendel's wants to remain together or tends to remain together during the formation of gametes, or when they like to cross over, or they like to more segregate, or like to transfer in a manner that is called non-parental type gametes. What are actually the parental type gametes? What are the non-parental type of gametes? Or what are the crossover type of gametes? Why the percentage of crossover or non-parental type gametes are increased? Why the parental type of gametes are increased? That we shall discuss in this chapter. So, let us start that what is first is actually the linkage or crossing over. So, linkage according to Morgan, linkage is Morgan worked with the Drosophila and the X chromosome linked genes. That is, he and his students observed that the genes or factors present on the chromosome tends to remain together during the transformation of gametes. During the formation of gametes, the genes or factors present on the same chromosome, here I am showing here the no crossover. Linkage is actually the phenomenon, it is the opposite of crossover, because the two factors or genes which are present on the same chromosome, they tend to remain together. This phenomenon is called linkage. So, linkage is a phenomenon in which two genes or factors that are present on a same chromosome, they tends to remain together, that is, they wants to remain together during the formation of gametes. So, and in this case, crossing over, what is crossing over? Crossing over where the genes present on the same chromosome are not tends to present on the same chromosome during the formation of gametes. That is, during the formation of gametes, they are transferred to the homologous part of another chromosome. So, I shall discuss in this matter, that is. So, let us start that when the crossover percentage is increased or when linkage is increased, when two genes on the same chromosome is present very closely. In this case, you see that two genes, that are R and Y genes, are present very closely on a chromosome. So, the tendency of these two genes to remain together is high, that is, linkage percentage is high, that is, crossing over percentage is low. When linkage percentage is high, crossing over percentage is low, and when crossing over percentage is high, linkage percentage is low. And in this case, the two genes distance are high, that is, two genes are placed distantly. So, why? The crossing over occurred between these two homologous chromosomes, in between two non-sister chromatids. These are called non-sister chromatids. So, when linkage percentage is high, crossing over percentage is low. When linkage percentage is low, crossing over percentage is high, and it is totally dependent on what? The distance between two genes present on a same chromosome. So, distance when high, there is a distance, and linkage is distance and linkage, distance and linkage linkage is inversely proportional. Inversely proportional. And distance and crossing over is directly proportional. That is, when distance is high, crossing over percentage is high. When distance is low, crossing over percentage is low. So, in this case, we have discussed about here the one parent, another parent. In this parent, what happened? Two genes are present very closely, and during the formation of gametes, these parents produce no crossover type gametes. That is, all are parental type gametes. Why? Because in this case, two genes are present on a very closely appraised condition, and in this case, two genes are present relatively at a distantly placed. So, the probability of crossing over between these two genes is high. So, in this case, what happened? First, in this case, there the chromosomes are present, there are two chromosomes. In this case, chromosomes are present, and after the DNA replication at the S phase, after the S phase is the G2 phase, and in G2 phase what happened? The chromosomes are replicated. Okay, chromosomes are replicated and chromatids are formed. So, these two sister chromatids are formed from each chromosome, and these two non-sister chromatids. These and these two non-sister chromatids, but these two are sister chromatids because they are formed from the same chromosome. So, when two chromatids form from the same chromosome called sister chromatids, and the two chromosomes form from and the two different chromatids of nearby chromosome called non-sister chromatids. So, in this case what happened? In anaphase 1, they are segregating, that is, first reduction division occurred. First reduction division, chromosome number 2, it is halved to chromosome number 1 in this case. So, no crossing over occurred because the two genes present very closely. So, no crossing over occurred in this case, and this type of gametes are produced. First the one cell is divided into 2, and then it is again after anaphase 2, each produce one gamete. So, in this type, this one produce this gamete, this one is going to this gamete, this is anaphase 2, the chromatids are separated like mitosis, and in this case 4 gametes are produced, and very carefully, look very carefully here, all gametes are of parental type, that is RY, small ry, though no intermediate type of gametes are produced here. So, no crossing over occurred here. So, when distance is low between the two genes on a same chromosome, the probability of developing the parental type of gametes is high. When distance is high between the two genes, the probability of non-crossover type, that is non-parental type, that is crossover type of gametes are produced. In this case, what happened? In this case, the same genes are present, but at a distant condition. So, why? At the Pachytene stage, at the Pachytene stage of prophase 1, prophase 1's Pachytene stage, the crossing over occurred between these two genes, and exchange occurred. What happened? In this case, the chromosomal part of this chromosomal part going to this is a non-sister chromatids. This chromosome replication occurred, and these non-sister chromatids exchange their material. We know that the crossing over occur between two non-sister chromatids, that is in between two non-sister chromatids of a homologous chromosome pair. So, in this case, this is a homologous chromosome pair, and crossing over occur between two non-sister chromatids. In this case, what happened? So, in this case, if we see that after anaphase, the same segregation occurred in this case, and after segregation, what happened? The gametes are formed after anaphase 2. In this case, what type of gametes are produced? These are the parental type. This one is parental type, capital R, capital Y, capital R, capital Y. This one is small r and capital Y, small r and capital Y. These two are non-parental type. Because this combination is not present in parent, that is small r, capital Y is not here. So, these are 50% gametes produced in this case are non-parental type, but in this case this is again parental type. So, when distance between the two genes is high, not only the parental type but also the intermediate type of gametes are produced, that are called crossover type of gametes and parental type. So, there is in this case one matter is very important, that when crossing over occurs between one non-sister chromatids to another non-sister chromatids, but remaining 50% chromatids of each chromosome not taking part generally in the crossing over phenomenon. So, what happened in this case? In this case 50% gametes obviously of a parental type, 50% gametes are of always parental type, and 50% gamete will be of crossover type, because these 50% not taking part in this chromatid, and this chromatid is not taking part in crossing over, but these two are taking part in crossing over. So, non-parental type of gamete and parental type of gamete are produced in a 50-50 manner. In this case, what happened according to Morgan? Morgan actually tested the yellow body and red eye and yellow body fruit fly with he crossed with, I have just make it clear that they have crossed with yellow body and red eye, yellow body with white eye, this one is mutated type, yellow body, Y for this is a yellow body and this is a white eye, and its parental type is wild type is, this is red eye, that is a red eye and gray body, this is a gray body and red eye. So, this combination, in this case one fruit fly, that is a male, this is one a female. In this case Morgan tested one female with female with a yellow body and white eye crossed with gray body and red eye, male, gray body and male eye. We know that in case of females 2 X chromosome are there. These are the 2 functional X chromosome, but in case of male 1 X chromosome is present, and another Y chromosome. We know that Y chromosome is not a homozygous, but is a hemizygous type with this. No genes are contributing, no counterpart gene is present here, that is no allele is present along with this. So, the Y chromosome is present but non-functional in that manner. So, when they cross together, what happened? F1 produced, first F1 produced. F1 what produced? This type of gametes, that is the male, produce the X chromosome. So, this X chromosome here, combination, this chromosome is coming here and this is another X chromosome here. And another Y chromosome is here and another type. So, there is only one type of gamete is produced, that is YW, because crossing over if occurred, it is a homozygous recessive type. So, there is no variation will be produced, only one type of gamete produced, because in this case Y and W, if crossing over occurred, what will produce? Y and W and Y and W. So, this is a yellow and white type. So, one counterpart of that is a homozygous recessive type, another is wild type. So, this F1 is, which phenotype will develop in this F1? That is there will be red eye, that is a wild type F1. This will be the wild type F1, all are wild type, that is red eye and gray body. This is the red eye and gray body. But in this case, the F1 is also male F1s. What will be there? They will yellow body and white eyes. So, if they cross again, all F1s are crossed together, then what will happen? Then what phenomenon will occur, we will discuss. In this case, that is, this one is a female. So, female will produce such kind of gametes. So, this one is a chromosome, this is a homologous chromosome. So, according to the rule of crossing over, this one first replicated, this will be first replicated. So, this one is replicated. In the same manner, this will replicated and to produce two chromatids, same centromere is there, okay, and this will again replicated with same chromatids, same centromere and 2 chromatids, and between these two non-sister chromatids, crossing over occurred, because in this case distance is high, and crossing over, after crossing over, what will develop? That is this one is okay, this one is okay, and here the crossing over occurs. So there's a one part, this part will attach with this part, and this part will attach with this part. So there is a reciprocal occurrence, reciprocal crossing over occur here, the exchange of the genetic material occurring. So exchange occur. So in this case as a hybrid, this chromatid will becomes hybrid and this chromatid will become hybrid, and exchange of genetic material occur. What exchange occurred? In this case Y plus, that is the dominant or wild type allele, is transferred to here, and this is a normal allele, mutated allele will go to this, attached to this chromatid, and as a result what will happen? The F1 gametes will produce of different type. These will be the parental types, this one and this one is unaffected. No genetic material exchange occur with these chromatids, but these two chromatids exchange genetic material. So 50% will produce what? 50% will produce non-parental type, and these 50% will produce parental type. So what are the different gametes? 1, 2, 3, 4 gametes, but in this case what happened? Only one X chromosome is there. Y chromosome is non-functional, so no crossing over occur between these two chromatids. As a result, Y chromosome, X chromosome contain Y and X, this is the recessive allele gene, and all the traits. So 50% traits are of this type. Same, that is the same type of allele is, the same type of allele is present, and same type of gametes are produced. So here only one type of gametes, one gametes of X chromosome containing, another gamete is only Y chromosome containing. So two types of gametes actually here, only one here, because this is the same, this is a copy of this, and this is another, this is a copy of this. So these are two type of gametes. So let us say checkerboard, if we form a checkerboard, then what will be the male, here the female will produce what kind of? 4 kind of gametes. What 4 kind of gametes? That is a wild type parental and recessive parental type. So recessive parental type, recessive wild type parental type, and a crossover type. 2 crossover type of genes are there. 2 crossover type, Y plus W and YW plus. No Y plus W and YW plus was there in a parental. So they are produced as a result of crossing over. So crossover type of gametes, and in this case only one type of gametes here, and another is Y chromosome, so non-functional. So as a result of these, we will see that 8 different type of phenotype in a progeny. What will happen? In this case, yellow, this one is yellow and white-eyed. This one is also yellow and white-eyed. This one is wild type, that is red-eyed and yellow body. This is a red eye and yellow body. This is a yellow body and what? Gray. Sorry, this is a red eye. This is a white eye and great. This is a white eye and red body. Sorry, white eye and gray body. And this is also a white eye and gray body. White eye and gray body. If W is a gray color, is determined. In this case, this is a wild type, that is the gray and red type. So this is a type of phenomenon Morgan observed. Now if the crossing over occur here, if the crossing over will not occur here, then crossover percentage will be of reduced. So you will see in NCERT that crossover type, when crossing over is higher, when distance between genes are higher on a chromosome, the crossover percentage is higher. When distance is low, crossover percentage is low. Thank you.